starting with Section 1, the Alzheimer's brain mystery, where we define our suspects.
Okay, so for over a century, literally since Alois Alzheimer first examined this disease under a microscope, two aberrant protein formations have been the undeniable hallmark signatures. Over on the left, we got suspect 1, amyloid-beta. This protein has a bad habit of forming clumps. It starts as small monomers and eventually builds up into these sticky plaques floating around in the extracellular space right outside the neurons. On the right, we have suspect 2, tau protein. Now, unlike amyloid, tau does its dirty work inside the cell, twisting itself up into neurofibrillary tangles. So remember that, outside versus inside, plaques versus tangles.
Let's look closer at Section 2, Suspect 1, Amyloid-beta.
We need to examine how a totally normal cellular component suddenly turns into a toxic threat. Amyloid-beta is actually just a small fragment. It gets snipped away from a much larger protein called the amyloid precursor protein, or APP, by specific enzymes. Normally, a short version of this fragment is cleared away, no problem. The brain just takes out the trash. But in Alzheimer's, a slightly longer, stickier version, usually A-beta 42, gets produced. Notice how those single monomers clump together into a group? We call that an oligomer. Now, for the longest time, we thought the final, massive plaques were the real killers. But science increasingly points to these intermediate oligomers as the main toxic species. They're small enough to remain soluble, meaning they can move around and actively disrupt the synapses.
And this brings us to the famous Amyloid Cascade Hypothesis. Think of this theory as a strict biological domino effect. It argues that the aggregation of amyloid beta is the primary, absolute trigger. This accumulation knocks over the first domino, setting off a deadly downstream cascade. It causes subtle synaptic injury, it triggers massive inflammatory responses from the brain's microglial cells, alters cellular homeostasis, and ultimately forces our second suspect, tau, to go rogue, leading to widespread cell death. It's a really compelling narrative, right? Amyloid pulls the trigger and everything else just follows.
So, moving to Section 3, evaluating the amyloid cascade.
As impartial scientific detectives, we've got to weigh the evidence. And honestly, the strongest evidence for suspect 1 comes straight from our DNA. There are these rare, early-onset familial forms of Alzheimer's disease. And here's the kicker. Every single genetic mutation that directly causes this early-onset form, specifically mutations in the APP, PS1, and PS2 genes, is directly linked to increasing the production or the stickiness of amyloid beta. It's a genetic smoking gun. Plus, we absolutely know that amyloid oligomers are incredibly potent when it comes to triggering a damaging microglial immune response in the brain.
But wait, the case isn't closed just yet. While those genetic links to familial Alzheimer's are pretty much undeniable, the clinical reality tells a much more complicated story. For decades, pharmaceutical companies have poured billions into designing drugs to clear amyloid plaques or stop the enzymes that produce them. And, historically, a lot of these clinical trials have yielded incredibly disappointing results or caused significant side effects, all while failing to meaningfully stop the actual cognitive decline in patients. So you have to ask, if amyloid is the sole kingpin, the big boss, why hasn't clearing it universally cured the disease? That very question has led a lot of researchers to pivot and look much closer at suspect 2.
Let's move to Section 4, Suspect 2, tau protein.
Let's shift our focus deep inside the neuron. So what exactly is tau? In a healthy, normal brain, tau is basically the cell's most reliable construction worker. Neurons have these incredibly long internal train tracks called microtubules, and they're used to transport nutrients and essential molecules all over the cell. Normal tau protein acts just like the structural ties holding those train tracks together, keeping the whole transport system totally stable and functional. But notice what happens when things go completely wrong. Normal tau usually has a very low level of a chemical tag called a phosphate group attached to it. But in an Alzheimer's brain, tau becomes what we call hyperphosphorylated, meaning it gets absolutely covered in these tags. Because of all that extra weight, it literally falls right off the microtubule train tracks. The tracks instantly collapse, which starves the neuron, while those detached tau proteins start clumping together. They form paired helical filaments and eventually build up into those massive, suffocating neurofibrillary tangles we mentioned earlier. Which brings us to Section 5, evaluating the tau hypothesis. Could tau actually be the true driver of the disease? Well, the tau hypothesis suggests exactly that these internal tangles are the primary cause of neuronal death, not just some downstream consequence of amyloid.
And the clinical evidence here is incredibly compelling. First off, human brain studies actually show that pathological tau lesions appear earlier than amyloid plaques. Second, we use a system called Braak staging, which maps exactly how tau tangles spread through the brain over time. And this spread perfectly, and I mean perfectly, mirrors the real-world clinical progression of a patient's cognitive decline. Essentially, where the tangles go, dementia follows. Finally, animal models engineered to express human mutant tau develop severe cognitive impairment, and if you treat that tau pathology, their performance actually improves.
So the crucial takeaway here is that tau correlates beautifully with dementia severity. But as always, there's a major catch. Unlike amyloid, there are absolutely zero known genetic mutations in the tau gene itself that directly cause Alzheimer's disease. Now, to be clear, mutations in the tau gene do cause other types of dementia, but not Alzheimer's. So it becomes really hard to argue that tau initiates the whole disease if genetic defects in tau don't actually trigger it in humans in the first place.
Let's wrap this up in Section 6, a complex interacting symphony.
Okay, so we've got strong genetic evidence for amyloid, but incredibly strong clinical correlation for tau. Which brings us to a really fascinating realization. What if looking for a single mastermind is just the wrong approach entirely? What's super striking here is the emerging parallel between our two suspects. For both amyloid on the outside and tau on the inside, modern science is increasingly pointing away from those massive, visible plaques and tangles as the true killers. Instead, for both proteins, it's those smaller, soluble oligomers that are highly toxic to the synapses. They're both basically operating through very similar patterns of misfolding and toxic aggregation. And this reveals the really modern view of the disease. It's not an either-or situation at all. The scientific literature now describes it as a cacophonous symphony. We know that amyloid oligomers can directly induce tau hyperphosphorylation, but how does it do it? Notice the middle of this graphic here. When amyloid injures the surrounding area, it activates the brain's immune cells, specifically the microglia and astrocytes. They start releasing these pro-inflammatory elements. This resulting neuroinflammation acts like a bridge, supercharging the toxicity of tau inside the cell, which then drives neuronal death. It's a bidirectional, deeply interacting web of toxicity. Which leaves us with a final, pretty profound question to ponder today. If Alzheimer's is indeed a cacophonous symphony, where amyloid, tau, and neuroinflammation are all continuously triggering and amplifying each other, will targeting just one suspect ever be enough? Perhaps the ultimate cure won't come from a single silver bullet, but from a combination therapy that untangles this entire biological web all at once. Thank you so much for joining me on this explainer. Keep questioning, keep learning, and I'll see you next time.